everyone. It's my pleasure to introduce Mathilde Meyenberg from Vienna. She's talking about obesity-associated cancers, a DNA repair perspective.
Thank you. So my name is Mathilde. I'm a PhD student in the Louisville lab, fourth year, and I'm standing in for my PI today. And I'm a wet lab person turned bioinformatician because my project got really data heavy. In my lab, we're focused on DNA repair and damage, and generally what impact this has on genome stability. And in my project specifically, I focused on obesity-associated cancers, and I'd like to give you a couple of highlights of our research. So through the previous session, we had a lot of introduction about exogenous, endogenous DNA damage, and that if this gets repaired properly with the appropriate pathway, we can upkeep genomic stability, and sometimes it's not. Either the cells die or they install mutations in the end. And today, I would like to focus on how we think that obesity might impact the balance between DNA damage and repair, and give two research highlights of cancers that we focus on, the esophageal cancer and colorectal cancer. For a really long time, like epidemiologically, it's become clear to the International Agency of Cancer Research that body fat accumulation does increase cancer risk, especially for organs along the gastrointestinal tract, and especially esophagus and colorectal cancers are two cancers which are very heavily affected by obesity, and therefore are a really good model to study how genome stability plays into that. But maybe we can take a couple of minutes to look at the background of this and how obesity increases cancer risk. And one of the prevailing hypotheses is that as adipose tissue expands, it takes on a sort of secretory phenotype where it secretes insulin, growth factors, hormones, and also cytokines, one of the prominent ones being IL-6. And all of these ligands then dock to their respective receptors and initiate a downstream signaling cascade, which allows cells to better survive, have a faster cell cycle progression, also be better at migration, and that this constant activation of these signaling pathways somehow predispose the cells in the respective tissues and lower the threshold for oncogenic transformation. And of course, if we have a high metabolic activation, then we also have increased ROS signaling, which can damage macromolecules. And there's also evidence of epigenetic dysregulation, which then in turn affects a lot of downstream processes. It sort of accelerates the whole process towards oncogenic transformation. And in my lab, in the project specifically, we are interested in how all of these changes in the obesity condition might impact directly or indirectly on the balance between DNA damage and repair. Because when this balance is disturbed, the cells have mutational outcomes, and these can be studied through the lens of mutational signatures. And I know this audience knows that you can put the mutations into their sequence context, and you end up with an overall profile, which is slotted into these 96 different mutation channels. And you also know that this overall lifetime portrait of mutations that a cell accumulated can be decomposed with non-negative matrix factorization, where the samples with their mutational profiles is basically a linear combination of the exposures that the samples received and the individual signatures that these can be decomposed into. And while this is a very mathematical construct, there is a point in the decomposition where it's stable. And studies which are recorded in the cosmic signature find like a host of signatures which are always extracted, which seem to be stable, and which therefore can be purported to have one specific biological process behind them. And we're interested in delineating these etiologies because that would allow us to understand A, how obesity impacts cancer risk, and B, how maybe cancers which are associated with obesity present differently molecularly, and what kind of impacts this might have for treatment. Because biologically speaking, to give a very powerful example, when you decompose the lifetime portrait into individual signatures and you know the etiology, this can guide treatment decisions. So I'm sure you heard of Hongmall's recombination deficiency, or the associated signature 3, which is this one with a very flat profile. And when you have this, then you have the so-called BRCA-ness phenotype, and anybody who has a defect in this process might benefit from PARP inhibitor treatments, because a defective homologous recombination is synthetic lethal with PARP inhibitors. And it's very important to know that these signatures are a result of a defective process, not necessarily of defective genes. So it is true that you can have mutations in the causative genes like BRCA1, BRCA2, or PAL2B, but it's also equally true that the signature can arise from epigenetic modifications or other modifications, including, for instance, an accumulation of 2-hydroxyglutarate, which just hinders the repair process. And these patients also have that phenotype and might benefit from PARP inhibitor treatment. Because if you just look for causing mutations, you would actually miss about 50% of the patient population that might benefit from PARP inhibitor treatment. And I just wanted to give this example to make clear why mutational signatures are clinically so important and why it would be so important to study them. And with that, I would like to jump into the first story, where I want to focus on obesity and genomic stability in esophageal cancer, which is a story that we've just recently submitted for publication. So esophageal cancer is not the most common cancer worldwide. It only lands on place 8, but it is a little bit of a silent killer because it lands on place 6 in terms of the mortality rate, because it tends to progress fairly symptom-free, and then patients present to the clinic at a very late stage of the disease, at which point treatment is a little bit less efficient. So the 5-year relative survival rate just falls short of 20%, and that is definitely something that should be improved. And furthermore, it's also one of the cancers most affected by obesity, because since the 1970s, we see a drastic increase, especially for esophageal adenocarcinoma, which is the subtype which is mostly associated with obesity. But we also see this crass discrepancy between males and females. But nonetheless, it is a very good model to study the effects of obesity on cancer development. And so we turned to publicly available data in the Cancer Genome Atlas, and our approach was to only consider early stage disease patients for 2 reasons. First, these tend to be resected before they receive treatment, and secondly, then we also exclude confounding factors that come from weight loss due to treatment or cancer-related cachexia. And when we took all of these different cancer types, here shown in the Y dimension, and plot their signature activity for high BMI versus low BMI patients, we see a selection of signatures that show a differential activity profile in the obese patients only. And of all of these cancers that we looked at, the esophageal cancer here, shortened with ESCA, seemed to have a very drastically different profile from normal weight patients, whereas we have a significant increase in the activity of signature 1, which is the canonical aging signature. We also have a surprisingly significant decrease of signature 16, which is a mismatch repair signature. And then we also have the increase of signature 17A and 17B.
So the bottom here is you're showing gene set enrichment analysis, and each column represents a different gene set?
No, the columns represent the activity values of the signatures, which are shown at the bottom.
Of mutational signatures?
Yes. So the activity of the mutational signatures.
And then, is BMI data available for all of these TCGA cases?
Yes, we only include those where we do have that body weight data available. And only those of the early disease, stages 1 and 2.
Corresponds to BMI? No, to the intensity of the signature.
Intensity of the signature in the individual cancers, but the difference between the obese versus the normals.
Okay.
Oh, that's right. Can I ask you, so when you subset by early stages, how many samples are you left with?
12 in the obese patients and 54 in the normal. And then we also have another weight category in between the sorry, I'll show this in a second. Here, so sorry, we have 50 normal, 25 overweight, 12 obese.
And that's across all cancers?
No, from here on out, I'm focusing on esophagus only.
But then you treat both squamous and adenocarcinoma together.
Yes, we do. We do. But we also do not find a bias by a histological subtype. We tested for that. So the obesity seems to be a stronger driver than the subtype. So in this But to convince us that basically the activity profiles are really different in obese patients, we also checked out this matrix basically with 3 different tools, so SINAR, Deconstruct 6, and mutational patterns. And here I'm showing for signature 17A and signature 17B the different cosine similarities in the activity results that we get. And they seem to agree fairly well, maybe with the exception of Deconstruct 6. But since we saw a somewhat linear increase in signature 17A and 17B for the esophagus cancer patients, and we see a significant increase in the signature activities when we compare normal and obese, we wanted to focus on those 2 groups and dig a little bit into what this means clinically. And these signatures are a little bit partners in crime. They tend to occur together. And we have signature 17A with the T2C mutations and signature 17B with the T2G mutations. And 17B was observed previously in combination with 5-fluorouracil treatment, but also both signatures tend to occur as a hallmark signatures of treatment-naive esophageal cancer, which applies to our data set. And that could basically indicate that the type of damage that 5-fluorouracil induce and how it's repaired might have something in common with the mutational process which are active in obese esophageal cancer patients. And because esophageal cancer development is tightly associated also with gastric reflux disease, it's been proposed that the resulting inflammation and the increase in ROS signaling from that could be a cause of these signatures, but we do not actually think that because we see an increase only in signatures 17A and B, not in the canonical signature 18, which is due to ROS, and 18 is mostly C2A mutations. So even if different ROS species and mechanisms generate the 17 signatures, one would expect that there would be a canonical increase with signature 18 also. And we do not observe that. In another study by Rebecca Fitzgerald's group, they found something interesting also that we wanted to follow up in, and that is that they could molecularly subtype different samples of esophageal cancer patients according to signature activity. And what they've noticed is that the patients where signature 17B and 17A were very active fall in a so-called mutagenic phenotype. So these patients tend to have an increased mutational load. And indeed, this was done from the pan-cancer data. And indeed, we find the same thing where we have a high signature 17A and 17B activity. We also have a significant increase in total number of mutations, so an increased mutational burden. And we also see a decreased survival chance, which is only significant for signature 17B. But since those 2 co-occur, that was quite interesting to us. When we look at the clinical impact of this and look at the hazard ratio for a lot of clinically assessed models, we see that only the 17B exposure levels are actually significant in terms of hazard ratio. And that kind of prompted us to ask, if this is so clinically relevant, like what kind of impact could this have for treatment? And it's been known in the literature that tumors which have a high mutational burden tend to respond fairly well to checkpoint inhibitor treatments. Yep.
Sorry. So it says BMI category here. It looks like overweight is protective and obese is, but none of these cross. They're both. No, they're not significant.
No, and that could also be an artifact because a certain overweight as you go into a disease state can be protective just in terms of energy balance, right? Like it's known that cancer patients, when they go on treatment, when they have a little bit more body weight to go in, they tend to fare better in terms of survival, which is not necessarily anything to do with the molecular presentation of the tumor. So anyways, since it's been observed that high mutation burden can be exploited potentially for checkpoint inhibitor treatment, the proposed model basically from the literature is that when cancer cells enter an accelerated cell cycle, they could become dependent on cell cycle checkpoints because they do incur still damage, and they need to repair this damage, and they need to have the cell cycle checkpoints to stop the cell cycle at certain points and give them time to repair themselves. And in that process, they become dependent specifically on WEE1 family members. And when you then add WEE inhibitors on top, which are currently in the clinic, these cells just can't deal with the damage anymore, are overwhelmed, and eventually that leads to mitotic catastrophe and cancer cell death. And this is just a model that we propose, but that still would need to be tested in the lab. But what we wanted to do is to say we have a clinical relevance of mutational signatures, but they're not very actionable if you have to go through this expensive process of sequencing and analyzing the mutational signatures for all of the patients. So maybe it's better to find a surrogate biomarker. And in order to find one, yeah?
So the relation between clinical benefit and the relation you explained before with BMI, do you think it's related or it's independent? With the signature 17, you showed it before related to BMI, right? And
yes.
Now related to a clinical benefit.
Yeah, I think it's related. So we see higher signature 17A and B in obese patients. And so that would be the patient population to focus on for stratification. They tend to have lower survival and higher increased mutational burden. And maybe they could respond better to checkpoint inhibitor treatment. I mean, that's something that we cannot test. So what we wanted to do to find a surrogate biomarker is to look at the available gene expression and methylation data and see if we can find commonly dysregulated pathways which are present in the obese patients only. And you can see the heat map for the gene expression on the left, and in the same order as the patients clustered on the right with the methylation, when we show the beta value of the promoter methylation. And it's obvious that the obese patients, even though we do have a low sample size, they cluster very separately from the normal patients, both in the gene expression and in the methylation data. So even at the early stages of disease, the tumor seems to present quite differently molecularly. And in terms of specific pathways, one of the most dysregulated what we found are genes which have to do with innate immune signaling. That are either significantly decreased in the case of IL-1 alpha here in the obese group, or in the case of, for instance, NOS2, very significantly increased in the obese group. And when we then correlate these genes, which are dysregulated also with BMI and the signatures, NOS2 is a gene which is significantly increased in the obese patients and also significantly correlated with the signature activity of both 17 signatures. For the anti-inflammatory genes, we see that potentially TGF-B1 and IL-10, which are decreased in the obese patients, they seem to have a sort of protective effect because they are, at least TGF-B1 is negatively correlated with the signature activity of 17. And when we look at the all of the genes which were strongly dysregulated, NOS2 is actually the top hit, which when we plot the expression of NOS2 also across different cancer types, we see that it seems to be a very tissue-specific thing in esophageal cancer. And so we wanted to focus on this gene a little bit as a potential biomarker, and it is an inducible nitric oxide synthetase, which takes L-arginine and converts it to L-citrulline with the toxic byproduct of nitric oxide, which also has biological functions, but the overproduction of this could potentially be toxic because it's a reactive nitrogen species.
One question as well.
Yeah?
So I don't exactly understand, why do you need a biomarker if it's already obesity that you have as a factor? So why don't you just look at obese people as the biomarker, basically, as BMI?
It wasn't just obese people that showed high signature activity, just mainly.
Okay, and so now you want to get also the other people that might have 17.
Yeah, yes. And also to dig into the pathway to see maybe what's related to the signature. But we don't think that NOS2 is causative of the signature because in Serena and Xanol's paper, the mutational companion paper, they also tried to expose iPSCs to nitric oxide specifically, and they couldn't recover a signature. Instead, it could be that nitric oxides damages like benzene-containing compounds, which are present as metabolites in the body, and then has an indirect effect on DNA damage. In any case, because we can model a linear relationship between expression of NOS2 and signature activities, both for 17A and 17B, it can be used as a surrogate biomarker with about 75% efficiency, 75% accuracy, which we think is quite interesting, and it could definitely be improved upon. But in summary, basically what we think we found is that we have the phenotypic presentation, which impacts on activity profiles of mutational signatures, and that has a direct impact on clinical parameters such as survival and tumor mutation burden. And with that, I would like to shift to the second part of the talk and present a little bit about our work of colorectal cancer, which is also an obesity-associated cancer that has a concerning increase in the trend that goes along with the obesity epidemic, especially in the young age groups, like 20 to 49 years of age. And with that cancer, we were more focused on wanting to understand what causes the increase in risk before the cancer even develops. And with that kind of question, it's quite difficult to use data which comes from patients who already have cancer. So instead, we wanted to set up a very clean bottom-up experimental system in which we can study the systemic changes that happen and characterize the genomic stability impacts. So what we did is we took wild-type C57BL6 mice, and we put them on a high-fat diet or a normal diet. And the high-fat diet, 60% of the calories come from saturated fat, and that is a diet which is sufficient to really just have a diet-induced obesity where we don't need to change anything about the genetic makeup of the mice. And we put them on a very long time course of this, and mice on a normal wild-type background, they tend to develop lesions more in the small intestine rather than the large intestine, which is a difference to the human model, which is why in our sequencing effort, we focused on the intestine and chose the organoid model to extract the intestinal stem cells, establish organoid culture, and derive clones from this. And we would sequence these clones from each individual mouse alongside with the tail DNA, so we could define which germline mutations each mouse has, even though it's a quite inbred strain, to then do the subtraction and only focus on the de novo mutations which arise in the organoid culture. And the genomic data analysis for us, we did in 2 stages. So once at the global level and once at the local level, and I just want to present a few highlights of there. So the results and at the global level, we employed de novo extraction with NMF and also signature activity tests where we map to the present cosmic signatures or different subsets of thereof. And at the global level, the signature calling didn't yield that many interesting results because we could extract 4 signatures. And as you can see, all of the signatures seem to have a very flat profile. There's nothing that distinguishes one over the other very much. When these are decomposed into known signatures, we could find 2 with a known etiology, so the aging or tobacco. And I know that my mice didn't smoke, so we think that the overwhelming C2A component, which is called as a tobacco signature, is actually an artifact from the culturing of the organoids in vitro. And we also could detect fairly flat signatures, 5, 8, and 40, which are unknown etiology, as well as some sequencing artifacts, which are unfortunately biologically not informative. At the mapping level, we could see that we basically do not have obesity-specific signatures, because what you see here is the activity levels of all of the of a subset of the cosmic signatures, and on the x-axis are the individual clones which were derived from the mice. We have the red ones from the high-fat diet mice and the blue ones from the normal diet mice. And I know there are a lot of plots on these slides, but basically there is no stark difference except for signature 55, which is known as a sequencing artifact. And when we dug into where those reads that were called with that signature actually came from, we verified that this was indeed a sequencing artifact. And when the analysis didn't yield specific results at the global level, we wanted to keep looking at the local level, where we defined specific genetic regions. Promoter regions, genic regions, and intergenic regions. And we looked at the signatures by the genomic regions, the mutations by the genomic regions, and the coding mutations as well. I will only focus on the mutations by genomic regions because the other two approaches didn't yield any interesting results. And one interesting thing that we found there was that when we look at CpG islands specifically, we see an increased mutational burden in the high-fat diet mice only. And this seems to be mostly driven by an accumulation of single nucleotide variants at CpG islands specifically. When we look at the composition of which type of single nucleotide variants are, the profile actually looks identical between high-fat and normal. So we are not sure how to interpret the increase in SNV, and we have to carefully look into how the changes at certain CpG islands might affect nearby genes, for instance, because CpGs tend to be associated with promoters. But basically, the negative results of the negative results in our genomics data tells us two different things. And the first is that there might not be any specific mutational signatures which are due to diet. And secondly, that the type of dysregulation that increases the cancer risk in a model of colorectal cancer has nothing to do with genetic regulation, but might happen in other realms of biological regulation. And in this project, we also collected other data sets through this time-resolved study design, basically, what we noticed is that, for instance, in the metabolome and the inflammation, also the microbiome, which I'm not going to show today, we noticed that there are occurrence of age-dependent changes, which we see normally happening from the first to the last time point in the normal diet mice. They seem to occur sooner in the high-fat diet mice. And that led us to this new hypothesis that we might have an increased aging process in the high-fat diet mice, and maybe that actually predisposes or increases the cancer risk.
Sorry, can I ask maybe an obvious question? Did those mice got cancer?
No, they did not.
They didn't, okay.
They did not. They would, without any predisposition or chemical intervention, you could keep them for 2 years, and they would, probably less than 10% of mice would develop intestinal lesions. Yep. So in the realm of metabolic regulation, for instance, we see that the diet-driven regulations seem to actually disappear at the latest time point. So at some point, the aging process seems to overwhelm any effects that the diet has. And that was quite interesting to us because basically that means the diet-dependent changes disappear, and that leads us to hypothesize that the early changes, which we see in the pentose phosphate pathway, ribose 5-phosphate, and so, which are diet-driven, they are also experienced by the normal mice as they age. And indeed, when we looked at this from a pathway level and compared the young mice and the old mice from the normal diet group, we see that purine metabolism, pyruvate metabolism, and the pentose phosphate pathway are the top dysregulated pathway, which just normally happen in aging. When we then compare the young high-fat diet mice versus the old high-fat diet mice, we also see the pentose phosphate pathway and purine metabolism being dysregulated, which is clear because they're aging. But when we compare the young normal mice versus the young high-fat diet mice, we also see pyruvate metabolism, and specifically the pentose phosphate pathway being one of the top most dysregulated pathways, which means that this is a age-dependent change, which we see, but it happens due to the diet. It happens sooner. And this is important potentially for genomic stability in the sense that the pentose phosphate pathway shown on the left here has this very important intermediate ribose phosphate, which was always depleted in the high-fat diet. And we think that is because it's being funneled off into purine de novo synthesis. So it's being converted through a series of steps into inosine monophosphate here at the top. And then this again is converted through a series of steps into GTP and ATP respectively. And if we expect a dysregulation in this pathway downstream here, we would also expect a dysregulation in the abundances of certain metabolites here, GTP, ATP, and also intermediates. And indeed, that's what we see. So starting at week 12, which is significant here, we have a depletion of guanosine, but an enrichment in adenine in the high-fat diet group only. And we also have an enrichment of the intermediates, which are before GTP, so xanthine and xanthosine, which are enriched even at the earliest time points in the high-fat diet only. And another realm of regulation is also innate immunity, which leads to a lot of inflammatory reactions. And when we explore the cytokines from the plasma samples, we can see the same trend as we see in the metabolomics. We have a distinct clustering of the diet groups here shown, the high-fat diet in blue and the normal diet in yellow in the early time points, but that difference seems to disappear as they age because there is an age-dependent dysregulation of immunity. Specifically, we observed that for GM-CSF, shown on the left here, where we have an age-dependent increase that appears sooner in the high-fat diet group, and also to some extent for IL-1 beta, which was quite intriguing to us. So last thing I want to show is when we propose that the high-fat diet mice have an accelerated aging phenotype, we would expect that to see that reflected at the genomic level at some point. And so we used Computel, which is a tool that indexes and aligns reads to telomeric regions to then estimate the mean telomere length. And we did this with our samples and saw an interesting decrease of telomeric length in the high-fat diet group, which still remains to be validated, but it is interesting to see that supporting our accelerated aging phenotype. Of course, we have to continue the story and also integrate it with our microbiome data, which I haven't shown here, but we would ideally like to characterize how obesity changes the aging parameters systemically and locally, and maybe also validate this in human data sets. So with that, I want to thank everybody in the lab and my supervisor, and I hope you enjoyed the talk, and I'm open to questions and feedback.
So this is a very basic question, but so you have sequencing problems with one of the batches, right? Which was the fat one.
And not the whole batch. So the signature 55, it was just that, if you remember the slide, there were a few clones that supported that, and those clones were exactly on one sequencing lane.
Yeah, I didn't understand the clone part really, what you meant by clones there. But
so for each mouse, we sequenced 4 organoid clones, which came from 4 different stem cells. Okay. So just to have more replicates.
So these are expanded, okay.
Yeah. So a few of the organoid clones, which were they were distributed across certain mice, they landed all on one specific lane.
So you use all the mutations or the clonal mutations?
Mostly the
expansion.
Mostly the clonal mutations. So we set the VAF cutoff between 0.3 and 0.7.
Because if you have a lot of these, you are removing reads in one of the classes, right? So you have less sequencing depth. If you don't normalize for that, that might explain the excess of mutations and the telomere.
We downsampled to the sample with the lowest sequencing coverage. So we aimed for 30x, and we ended up with 28x in the end. Yeah.
And then with the TCGA and the BMI data, did you have data, were you able to correct for tobacco and alcohol? It was also because you got that signature later on when you were looking at the mice, so I was just wondering, can you actually pull that apart given that those are potentially all confounded?
Yes, I mean, that's always a problem with the clinical data. We haven't looked at that specifically. That's a fairly good suggestion. With what I said about the tobacco in my mice, like I know this for sure because I handled the samples, so I know it must have come from a culturing artifact. But in regards to the esophagus data, we'll have to look into this. And we'll also have to look into it with the pan-cancer data set. But it was encouraging that in the literature, they saw also this hypermutator phenotype in esophageal cancer patients which have these signatures active.
There's an old data set as well, which was pan-cancer data set, which had a ton of BMI data.
Mm-hmm.
I'll get you the reference on it. It's got like hundreds of patients in it. But because it kind of preceded the TCGA by maybe a year, it was eclipsed by it entirely.
Mm-hmm.
But it does have a lot of BMI data in that.
Yeah, that would be interesting. Thank you.
I can't remember the other thing about it may be thinking about IL-1 beta, leptin and everything. You've got deregulation of the HPA axis there. So all of that is all interconnected. So there's actually a lot of work in that field. Kath Lawrence has a lot of stuff in that area.
Mm-hmm.
She's in Manchester.
Thank you.
Now, this part of this association between BMI and signature 17, I think is very, very interesting. And then you talk about this high-fat diet associated with purine levels.
With what?
This may have something to do in relation one to another, because in signature 17, at least when we see it associated to 5-fluorouracil, this is also related to changes in the pool of purines.
Mm-hmm.
Because 5-fluorouracil blocks the synthesis of thymines, and then this ends up leading to this T2G mutation pattern.
That's very interesting. I hadn't considered that connection because we specifically didn't see anything in the mouse model. Right? Of course, mouse and human is not the same. I just never. That's a very interesting suggestion.
I don't know if it may have something to, but at least it rings the bell that it may be connected.
Mm-hmm.
Sorry to insist on that, but for tobacco signature, like you said, it can be a culture technical artifact that I noticed. Like I first thought, did someone smoke under the hood? Like how do you
No, it's just the C2A with a tobacco smoke. So it's like the same signature can occur in different contexts due to different etiologies, just because a certain type of damage might be very similar in evoking these C2A mutations. And that damage is happening when somebody smokes and it's seen in lung tissue, but that damage also happens when you have reactive oxygen species just damaging the DNA. And as you handle these tissue-like organoids for a month or so until you generate your clones, then you can expect that this is not a normal setting for them. And this is why we propose that these abnormally high C2A mutations we see actually come from that context.
Just a very simple question, because I don't deal with the signatures. Are they universal between organisms, generally?
I mean, that's a very hard question to answer because the same signature can happen in different contexts. So they could also be different in different organisms. What I mean by that, for instance, the UV signature, 7 is commonly seen in skin cancer because it makes sense that the skin is exposed to UV, but signature 7 has also been found in neurons, and we would not expect our brain to be exposed to UV. Instead, the signature came about from reactive aldehydes, basically, that induce a similar kind of damage, which is being repaired by a similar kind of process, and make the same signature, but from a different cause in a different context.
Yes, I understand that, but my question is about different species, because different species can, for example, have different DNA. So maybe it's not possible to get an increase in this type of mutation because it's just like those letters they appear much more frequently or less frequently, and I think that mice, like, they tend to live shorter because they don't like protect their DNA. Their DNA damage repair is not that active. That's what I've heard, because if you like let a mouse live like a good standard life, they tend to get cancer much faster than humans, right?
Yes, but I mean, they also tend to get different kinds of cancers. They have different kinds of exposures. It's not a perfect model, but in general, DNA repair is fairly conserved. And when mice are treated a certain way, their DNA repair systems tends to react a certain way, even though there might be different kinetics. So you're right. There can be underlying processes that change the outcome on the mutational spectrum, but you have to work with the model that you do have.
I just wanted to ask.
Yeah.
Why did you work with clones?
Sorry?
Why did you sequence clones? What's the purpose of that?
The purpose.
You could have sequenced a piece of tissue, no?
Yeah.
I mean, tissue. You have. So if I remember the slide, so you get the mouse and then you span 4 stem cells into 4 clones.
Yes.
Why don't you do standard sequence, like bulk sequencing of millions of cells?
Yeah.
So at the end you're representing 4 cells, right, of the mouse?
Yes. Yes. Exactly.
So there's going to be a reason for this, no? Because that's unusual.
Yes, it's actually the gold standard in the literature for this bottom-up mutational signature generation, because it makes the mutation calling that much more secure, that you know this is that mutation. Otherwise, when you have
To distinguish errors in the sequence.
yes, exactly. I mean, if you have a tumor and you sequence that bulk, then you already had a process in vivo of clonal expansion. It's still heterogeneous, but you have pockets of clones which will enrich the sequences, the reads that you have, and makes it possible to call which mutation is which with a given certainty. With this model, we wanted to make sure that we can call mutations with a given certainty rather than sequencing bulk, because most of the surrounding tissue would be normal, and we were afraid that there would be like too many.
On the other hand, you are just sequencing 4 cells, right?
I know.
Something happened to this cell by
Yes,
anything, it will distort a lot. Why?
Yes, and there's also the danger of a bottleneck of cells which already have a selective advantage somehow growing out better in culture. This was a decision in balance between biological interest and also, we cannot sequence 100,000 clones.
No, I understand, but you could infer signatures from bulk, no? That's the common strategy.
About normal tissues, David, she wouldn't see anything. I mean, she will
Signatures.
have a lot of mutations in individual cells or in small clones, and you can't detect this. Like sequencing is not a in vivo clonal expansion due to tumor. These are perfectly normal tissues.
I mean, there are a bunch of papers sequencing, in human somatic,
Either laser microscopy
sequencing 200 cells, with
No.
laser capture microscopy.
Yeah,
I mean, all the
this was
welcome
another
campus.
approach. But there
And you
is
see
also
signatures there.
But in the laser capture,
amplification of clones, so it's an alternative model.
but in the laser capture, it also assumes a certain clonality. If you think of the tissue architecture in the gut, then you would assume that the crypt or maybe any neighboring crypts actually might be
You can check
related.
in the buff. You have to look at the buff, you see you have 1 clone or more.
Yeah.
If the buff is like this, you have 1 clone around 5.5.
Yeah.
Yeah. Okay.
All right, I think it's time to move on. If there are no more questions, then let's thank the speaker again.
Thank you.